Dr. Vijender.
A very good evening to all of you, and welcome to the 18th official webinar of the IAMM-TAPC. Today we have one of my favorite faculty, Prashanti ma'am. She is the senior professor at and head of department at Siddhartha Medical College. And today, madam is going to speak to us about anaerobic bacteriology, which is going to be very useful for all of us. We welcome you, Prashanti, madam, and thank you for agreeing to take this lecture. Over to you, ma'am.
Thank you, Dr. Vijender, and thank you one and all. And at the outset, I would like to thank the executive body of IAMM-TAPC who is conducting these webinars and which are really very useful for all the microbiologists, both the faculty, seniors, juniors, and the young postgraduates and everyone. So thank you for the opportunity. And actually, I think I'm audible.
Yes, ma'am. Yes, ma'am.
Yeah, thank you. So this particular topic, I'll just share the. So is the screen visible?
Yes, ma'am.
Yes. So anaerobic bacteriology revisited and strengthening the anaerobic culture practices. This is actually a day-old topic, and which is I don't know how far it is important, but yes, it is. We believe that it is very important. Why I have chosen this topic? First of all, I want to uh, clarify, because most of these areas we just go through the books and we read and we just obtain knowledge from various sources. So in fact, the practicality of these areas are a bit lagging behind for even though in all these years of our working in microbiology. But fortunately, I has an opportunity to closely observe these anaerobic workups and how important it is and what is the meticulous nature of the work in that. So while I was working in Gandhi Medical College, and our postgraduate at that time, Dr. Vishwanath, so now he is a consultant at Hyderabad. So he has taken up this work, and we are really overwhelmed seeing his work, because he has taken it and he did it very committed and pin to pin. And so how important this taking up anaerobic cultures, not at the laboratory side but at the hospital side. So seeing him, we closely observing him, we develop a lot of awareness or knowledge on these areas. So since then, I am keen in taking up this topic, in sharing my knowledge to the youngsters. So now let us see the objectives. So to review the history and evolution of the anaerobes, so to understand the clinical relevance and what are all the updates in anaerobic culture procedures and the importance of the culture practices. So we all know that living organisms began to evolve in warm oceans. Those millions of years ago, free atmospheric oxygen hardly did not exist, and all the molecular oxygen was bound in various sources like water, carbon dioxide, carbonates, and sulfonates. So the entire planet was over a time filled with anaerobes in those days, and the evolving microorganisms has no choice but choosing a anaerobic pathways. So that way, anaerobes were the leaders or the beginners of the universe, we can say. And come to the historical aspects. So Anthony van Leeuwenhoek, so even though he was not a scientist, but his keen, enthusiastic observations has invented in so many ways. So his animalcules, while he was observing, he found that some of them, the organisms are live and mobile, even in the absence of air. So this was the first evidence which he reported in his document to the Pasteur Royal Society of Scientists or London. And later, Louis Pasteur further clarified this anaerobic workout. So in 1861, his observation was Vibrio butyric, a Clostridium butyricum. So in the wet mount preparations, it is losing motility when it is exposed to the air at the edges. So over time, Pasteur coined the term anaerobes for these particular organisms, which is the origin for the term anaerobes. So we all know that anaerobic bacteria are everywhere in the soil, on the humans, animals, and in the human beings, almost all the mucosal membranes are occupied by the anaerobic bacteria, especially the oral cavity, gastrointestinal tract, and female genital tract. So the ratio of anaerobes to aerobes is 1,000:1. It's a wide range, if at all we observe. And all these anaerobes, most of these anaerobic bacteria are encountered in infections in the human body involving almost every organ or anatomic site. And still, even though they are causative for various infections, establishing anaerobes as the contributing factor for the infections is really difficult or problematic, because most of these anaerobic infections are a mixture of both anaerobes and facultative anaerobes and part of aerobes. And being a part of the normal flora, most of these sites are contaminated with the normal flora, means the specimens are contaminated with the normal flora. Hence, assigning the clinical significance, even though we isolate in the laboratory, is a challenging. So come to the various diagnostic challenges. So actually speaking, anaerobic workups has become an endangered areas. But still we need to work out on this. And most of the labs, even maybe a tier 1 labs also think that it is a expandable luxury rather than a necessity for the diagnostics. Because again, the same scenario, the polymicrobial nature, most of the normal flora is contaminated in the specimens, and meticulous way of collection, transport, handling, which is really difficult to monitor, to educate from the site of collection till it reaches the laboratory. And what are all the methods, the conventional methods which are being practiced are costly and time consuming. And even many of the laboratories are not capable of establishing this, always making it available. And moreover, the clinicians are not much keen about our reports or our results, because the presumptive identification itself suffices for them to initiate the empirical treatment. So come to the trends of infectious prevalence of anaerobes. In 1970s, the anaerobic infections are accounted for 11 to 27% in most of the blood cultures. And when it comes to the 80s, the incidence has decreased by 45%. So this is really very good sign, but the possible reasons are due to improved surgical techniques, the bowel preparations and all that are increased, because the bowels are lower, large intestines are mostly filled with the anaerobes. And again, the diagnosis of infections, the early and accurate diagnosis and the methodologies are increased. So the availability of investigations is improved, and also use of broad spectrum antibiotics, which most of the times covering the anaerobic bacteria infections also. So because of this, the probable reason that the infections have come down in 80s. But again, in 90s to 2000, there is an acute enhancement of 74%. May not be acute. In this, over a period, it was observed that 74% of increase in the anaerobic bacteremias, because the aging population is increased, and immunosuppressive drugs are more in use because the cancer treatments are more evolved and transplantation surgeries are more undergoing. and even the evolving of the antibiotic resistance patterns. So these are all various observations from the literature. And actually speaking, between 60s and 70s, it is known as the golden age of anaerobic bacteriology, because Sotar, Feingold from Los Angeles, and Dowell in Atlanta, and Hodelman and Moore in Virginia. So they have done extensive work in that particular decade and expanding our clinical and laboratory knowledge of anaerobic infections. Their contribution is tremendous in that period. So there is a rising importance in the diagnosis of anaerobic infections or anaerobic bacteria from the clinical materials, because there is an increased morbidity, mortality associated with anaerobic infections, and also improved laboratory recovery by provision of various automated systems and several other areas, and more number of clinical labs are working on this. And overall the cost is coming down, and the automation is really useful because the TAT is come down, and there is a really practical to perform these, to take up these works. And apart from this, so it is not that simple. We have a sophisticated laboratory and sophisticated equipment, because it is not sufficient to diagnose to diagnose the anaerobes as the agents of infection in a particular individuals or patients, because again repeatedly we come across the word that they are the part of the normal flora. So polymicrobial nature of the clinical specimens is very, very important. So simple presence of anaerobes in your laboratory isolates is not a mere meaning that they are the cause for agents. So there should be some clinical evidence suggestive of anaerobic infections. So always obtain this history from the patient side by the clinician. So there should be a foul smelling discharge or a putrid discharge from the patient, the area or the infected site, and abscess formation. More of the closed abscesses are by the anaerobic infections, and tissue necrosis or any kind of gangrene or involvement of nearby mucosal surfaces, and the infections which are failed to respond to the routinely given antibiotics, which usually kill the aerobes. So this provide a clue for us whether the possibility of the anaerobic infections. And again, so accurate or proper sample collection and transport is very, very crucial for anaerobic workup to take up. So always prefer to collect the samples from the deep tissues, pus samples, closed abscesses, drains, and normally sterile sites if they are infected. So obtain samples from these under strict aseptic conditions. And consider most of the samples which are close to the mucosal sites and contaminated with the materials. Usually those kind of samples are not accepted in the laboratory unless otherwise there are any specific recommendations from the clinician after with the thorough discussion. Okay. And whatever most of the aspirates we do in the syringes, and see, once we collect, so even look that all the air pockets from the syringes are expelled, the bubbles or any air pockets are expelled, and remove the needles and seal the hub either with a parafilm or any caps. Even nowadays, commercial syringes with a hub caps, sealed caps are also available in the market. And always choose the anaerobic transport media or tubes or vials. Without that, never accept any sample received in the routine sample containers mentioning with a statement that anaerobic culture. It is totally useless, and you will never isolate anything, and it is a totally misleading for the diagnosis also. So never accept any kind of gauze from the tissues which are dressing gauze or cotton swabs, because they tend to trap the air, and the fatty acids present in that inhibit the growth of the anaerobes. And all the materials, clinical materials or specimens collected, see that even though in an anaerobic transport media, they should be transported as early as possible, preferably within 30 minutes. In case of delay, maximum Like any other, our routine, also we mentioned the same maximum delay of 2 hours. And do not refrigerate the samples meant for anaerobic cultures because cold damages some of the obligate anaerobes. And along with this, always obtain the definitive clinical data like site of infection, any prior antibiotic usage, is there any foul odor discharge or any necrosis or gangrene, etc., or presence of any gas. So once you receive the materials, clinical material, the specimens, so even inoculation is also very, very important. So here in the anaerobic, we can find out starting from the pre-analytical area, which starts at the patient site, from the collection, how to pick up the sample, how to transport, and how to come to the analytical area within the laboratory. Everywhere there is a meticulous way, there is a meticulous focus, and or media or everything should be focused and meticulously done. So here the primary media to be selected, always choose the freshly prepared ones, and or maximum of 2 weeks. So whatever media you prepare, use within 2 weeks. Long-term storage of media accumulate peroxides, and these peroxides inhibit the growth of the anaerobes. So it is ideal to prefer PRAS media, which is pre-reduced anaerobically sterilized media. And these media are now widely used and available commercially. They eliminate the dissolved oxygen, and they have a shelf life of 6 months. And so at every level, for every sample, whatever you receive, once you inoculate, definitely you make a gram stain and observe what are all the various types of organisms present and what are all the inflammatory cells present and how many number of organisms present, and all that you record and keep it for correlation with the cultures. So the primary plating always prefer a wide variety of media for anaerobes, because again, remember, it is polymicrobial because there is every chance of contamination with the normal flora. So select one non-selective anaerobic blood agar and one or more selective medias, and again 5% blood agar or chocolate agar and a McConkey agar, because being a polymicrobial, we want to rule out the aerobes, facultative anaerobes, and obligate anaerobes. And along with that, always make a backup broth with the thioglycolate broth. So this thioglycolate broth helps in enriching the small number of anaerobes if present in the clinical specimen. So all this process of inoculation should complete in a less than 15 minutes. It's such a quick process. It should be done in a 15 minutes time, because the duration of exposure to the atmospheric oxygen or to the surrounding air will kill most of the anaerobes. So that particular section we have to take care of. That process we have to take care of. So whatever the process done, immediately incubate at 35 to 37 degrees centigrade, and preliminary observation till 48 hours. And always the incubation should be in the anaerobic atmosphere only. So prefer either anaerobic jars or anaerobic pouches or chambers. So these are all the various anaerobic media, the selective and differential media like Bacteroides bilescline agar, laked kanamycin vancomycin agar, and anaerobic phenyl ethyl alcohol agars, cycloserine, cefoxitin, fructose agar. So each one, depending on the site of infection and the suspected pathogen, we have to select. So, and various non-selective media are also there: anaerobic blood agar, egg yolk agar, peptone yeast extract glucose broth, PYG broth, and cooked meat broth, and thioglycolate broth. Usually these broths help both in the cultivation and backup broths, and also if at all future testing for gas liquid chromatography. So this is another area. Metronidazole disc, 5 micrograms disc, helps or plays a key role in the identification or the preliminary identification of anaerobes in the culture media, because they help to differentiate anaerobes from the aerobes and also to guide the clinician in the empirical therapy. So these metronidazole antibiotic reduced in the anaerobic environment and release the free radicals, which damages the bacterial DNA and kill the organisms. So more than 15 millimeters zone, if we observe, so then it is susceptible. So then in the usually this metronidazole disc we place in the primary streak lines. So if there is a zone of inhibition of more than 15 millimeters, then you can suspect the organism could be a anaerobic pathogen. So there are further clues required in the culture, because once you have inoculated, after 48 hours, you want to observe for the any growth present in the culture media. So at that time also you need to focus on when you open the pouches or jars, observe for any foul odor when you open the jars. So that again indicates that the possibility of anaerobic organisms. And again, while reading the plates, observe for colony types, how many types are there, and compare with the other plates in other aerobic and facultative anaerobic plates. So what are the morphotypes or colony types? So, and which type of growth is seen, which selective media, and is there any pigment production? Is there any fluorescence observed? So all these factors we have to observe, which helps in the identification or confirmation of the anaerobic pathogen. So while observing or while recording the primary plates, always record the level of growth or the amount of growth. Is it a light growth or moderate growth or heavy growth, or even some labs may adapt as 1+, 2+, 3+, 4+ growth. And along with that, even your size, shape, edge, color, opacity, pigment, hemolysis, is the colonies are pitting or any, all these points are important to come to a final conclusion of the pathogen. So when you look for the yields, as I said, all the selective medias are meant for presence of any anaerobes or what are the various types of anaerobes. So all the colony morphologies are very important. Even in your non-selective media, if you find various colony morphologies, even for purification, you can subculture them into, again, to purity plates, either blood agar, anaerobic blood agars, or even a selective agar also you can prefer. And simultaneously, don't forget to examine the thioglycolate broth, which is a backup broth. So if any turbidity is observed in the thioglycolate broth, perform a gram stain and again see in the gram stain what are all the cellular types found. And if thioglycolate broth shows any cellular types which are not found in the primary plates already incubated for 48 hours, then you can subculture from this particular thioglycolate broth. So this again, you have to keep in mind as a backup broth. So if any plates or thioglycolate, no growth observed for 48 hours, but still you have to continue incubation for 5 more days. And after 5 days only, you can discard the material. And thioglycolate broth also usually 5 to 7 days is recommended. So once the colonies are seen, you can perform the grams. So this is like our regular culture phenomena, but only thing, the selection of media and the work duration should be minimized, because your idea is to minimize the exposure to the air, because most of the anaerobes get killed at that particular exposure. So observe the gram stain and various colony types, and wherever required you subculture. You can either incubate in the anaerobic environment, or even you can test for the aero tolerance by CO2 incubation. And so any colony morphology, you are the growth, amount of growth, and the specific conditions met as a suspected anaerobe, you can make further streaking into the anaerobic blood agar. So first and second quadrants, as much as possible, you take a heavy growth, and other quadrants for look for the isolated colonies. It is not like our floral culture, like it is like a lawn culture. We have to put 2 quadrants heavily and the remaining quadrants lightly, so that we can look for the individual colonies. And in the primary streaks, always try to use the potency disc like vancomycin, kanamycin, colistin with the specified concentrations, because these help to for the preliminary identification of the anaerobic groups like gram positive, gram negative. So gram positive organisms are sensitive to vancomycin and resistant to colistin. Gram negative are resistant to vancomycin disc. So So along with this, the other factors which we already mentioned, aerotolerance, colony, hemolysis, pigment, etc. So the potency of the various discs is given in a tabular form for which bacteria is producing. I'm not going to into the details of this. So these are all the various anaerobic bacteria of medical importance. The most important is the Bacteroides species, which is a more prevalent normal flora in the large intestines of the humans and also excreted in the feces, and it is present always. And so this is a very well studied and well known anaerobic pathogen because it is known for the development of resistance to antibiotics, and it has several virulence factors, so various endotoxins, capsules, enzymes, etc., and causes various tissue necrosis and abscesses. And Prevotella parfiremona species, and again they have a specific Prevotella especially produce a pigment, iron protoporphyrin pigment. So and even capsular polysaccharides, Fusobacterium. It is easy to identify Fusobacterium in the colonies because it produces a breadcrumb-like colonies, and even there a butyric acid pungent odor, a butter odor of butyric acid. And Propionibacterium, which is a pathogen in the acne, rarely causes endocarditis, and it is a partially aerotolerant also. So we can means if it partial exposure, it won't kill easily. And Actinomyces species, they are gram-positive bacilli, long and branched. So the typical growth is a molar tooth appearance. And Clostridium difficile, it is a more important nowadays because of its antibiotic-associated diarrhea or pseudomembranous colitis. So come to the various media. So anaerobic transport media, which already I mentioned. So only thing I want to mention here is nowadays the ready-made tubes are very much available. Even the tubes, empty tubes, where we can prepare our own media and fill are available. They are called as Hungate tubes, and with a which are fitted with a rubber septa. So no need of opening the lid. We can directly inject the clinical material into that. So they obviously have the minerals, buffered mineral salts, and reducing substances like thioglycolate and cysteine. It is in the semi-solid media. So they maintain the viability and also nullifies the lethal effects of the oxygen. So these are all a list of culture media, both liquid and solid, or the agar plates. So selective and non-selective. So the bolded ones are the selective media, the Bacteroides bilescline, Laced kanamycin, vancomycin, phenylethyl alcohol agar, and CCFA, cycloserine, cefoxytin, fructose agar. And the rest are the non-selective media, which are preferred depending on the type of sample or the suspected pathogen. So moreover, any kind of media used for anaerobic processing, either it's a transport or the solid media, the basic composition includes a dextrose, it provides the energy source, peptones are the nitrogenous suppliers, and the yeast as a vitamin supplement provides vitamin supplement. Sodium chloride is essential electrolytes, and cysteine and sodium bisulfite, reducing substances which maintains the low pH. Shea blood added for demonstration of hemolytic properties, and hemin and vitamin K are the growth factors. Vancomycin inhibits gram-positive, kanamycin, gentamicin inhibits gram-negative. So these are all various medias, just the picture collected from the internet. So once the media, we have known the basic processing now. So what are all the media used now? Again, I mentioned the entire processing should take place in the anaerobic environment only. So how to provide the anaerobic environment for the culture media during incubation? There are three major mechanisms: the jar system, pouch system, and the glove box system. So in the jar, it is an evacuation replacement method. So the inside air is removed and other gases are filled. And whereas pouch is a chemical method, the chemicals absorb the oxygen and release the other like hydrogen and carbon dioxide. And glove box is a closed system. The entire chamber is supplied with the anaerobic environment, provided anaerobic environment. There is a continuous monitoring and supply of gases. So it is a more sophisticated and controlled system. So this tabular form gives much more details. So there are three types of anaerobic jars: the gas pack anaerobic jar, Macintosh jar, and the anoxomat system. So all the jars, most of the they are made up of polymetacarbonates, and they are provided with a pressure gauge and a safety valve and shredders, inlet and outlet valves for the evacuation and pumping of gases, a cold catalyst, and a carrier for the Petri plates. So this cold catalyst means it is a palladium coated pellets, and they remove oxygen by reacting with the hydrogen and and produce the water. So in a form of moisture, there won't be huge collection. There are few drops or a moisture like water is produced. And so how do we know whether anaerobic environment is achieved or initiated or not? So we can observe there is a development of heat within minutes. So if we touch the jar or if you touch the pouch, you can see the heat generation. And slowly there, in case of pouch, there is a collapse of the cover, more tight collapse of the pouch we can observe. And there is a moisture also in the jars. So if you observe, there is a small moisture gathering on the jars and heat generation and slow collapse in case of pouches we can observe. And so this entire reduced conditions are achieved in 1 to 2 hours, even though methylene blue or resazurin indicators are there, because they takes little longer time to show the reaction. So the same anaerobic jar systems, all the three with a tabular form with a few more points added. So these are all the jars. So the gas pack jar, and this is the safety valve and the traditional Macintosh fill dish jar, and an oxomat jar where there is a computer operated database. So it helps in the maintenance of the computer operated, the gas controlled system. So everything is under the control of the equipment. So it is a more sophisticated, a bit costlier, but more useful. So next to the jars, there are anaerobic pouches are available. And actually, this pouches is a much easier system, which I practically experienced in working out with this. Because these are the sealable plastic pouches, and we can completely evacuate the air by simple pressing with the hands, and there are clamps provided. You can tightly clamp them. And so along with the inoculated plates, you have to keep inside the gas generating sachets and the indicator strips. So then seal tightly and incubate at 37. So the inner chamber, the gas packet releases various gases, hydrogen and carbon dioxide. The hydrogen absorbs the oxygen and releases the water. So little moisture developed, and there is a heat generation we can observe. So the advantages of these pouches is, even a small setup of labs, this is very useful. And it is useful to, if any bedside sample collection or inoculation is there, when you are not ready with the 100% anaerobic media like PRAS media, even your blood agar also works at the bedside to collect the sample, directly inoculate, and put it in the anaerobic pouch and transport to the lab. So this is a quick process, and even you can use as a transport media. Another advantage is we can visualize the plates physically through the pouch. Without opening the pouch, you can look the plates whether any growth is there or not. So, and another thing is they are disposable, portable, no electricity needed, anytime you can process. So only thing is maintaining the air tight and removing the complete air is important. So these are all various pouches, suppliers, means the market available. Standard anaerobic pouch and BD gas pack, EZ pouch, and biobag environmental pouch. These are all various pouches. So, and with the various sachets we have to put, and so it's just a tabulated. And again, so we need a gas generating sachets where they provide the chemicals present in that, the reactions provide the anaerobic environment. So there are different gas pack. The traditional gas pack, which contains the sodium borohydride and sodium bicarbonates. And there is another gas pack by the BD, which is a hydrogen-free gas generating system. Those chemicals, they do not produce the hydrogen. So the oxygen is absorbed by the iron-based chemicals that are present in the sachet. And the CO2 generated is activated by humidity and maintain the environment. So in this, there is no particular catalyst is required. The third category is the anaerobic pouch system, almost similar to the traditional gas pack system. But here, only dry chemicals are activated once they are exposed to the air. So we have to cut the sachet, and there is a absorbable, porous cover. Through that, the chemicals get exposed to the air. So these are all various anaerobic pouches with gas packs inside. So, and every anaerobic system, whether it is a jar or a chamber or a pouch, we need to provide one catalyst and an indicator. Catalyst is mandatory, and indicator may or may not require, depending on the type of system you have chosen. So the catalyst basically removes the oxygen, it catalyzes the reaction and removes the oxygen and maintains the anaerobic environment. So, and it is an active reaction. So always a chemical reaction which is active, we can observe whether the oxygen is reduced or not. And here the commonest catalyst, catalyst used is the palladium, and it should be always maintained dry, and any kind of moistures inactivated. And even after use also it remains unchanged, but it gets inactivated. To reactivate it, it has to be heated at 160 to 180 degrees centigrade. So palladium catalyst is a reusable material. And come to the indicators. Indicators are a passive methods, and they detect only whether oxygen present or not. So, and if the indicator get exposed to the oxygen, they get oxidized and change the color. If they are in the reduced state, they remain colorless. The commonest indicators used are methylene blue and rizazorin. So these are the palladium catalyst provided in a sachet, a small sachet, and methylene blue indicator. And come to the anaerobic glove boxes, and everybody I think knew about this. So it is a sophisticated equipment, and a clear plastic big chambers with airlock system, and the pass boxes are also controlled with the anaerobic environment. We can use the gloves which are airtight around the hands, and no exposure to the oxygen. So all elements are well maintained in the anaerobic environment. There is a continuous air anaerobic gases mixture flow throughout the chamber. So these are the chambers, and working for the anaerobic samples. So come to the automation role in the identification of anaerobes. So this table is a overview, but here I wanted everyone to see the point observed is all these automations help in reducing your workout of the biochemicals, which is a studious procedure, and takes a long time, and any point of contamination will give erroneous results. So, but even automation also, you need in hand the specimen, the cultured isolate should be there in your hand. Then only you can proceed further. So direct samples nowhere can be applied for the detection of the pathogens. So all the API systems, micro scan, walkaway, rapid ANA, REML. So all these are there. And nowadays Malditoff has come up, which is very crucial because it gives the fastest even 15 minutes ID. But provided again, you need a culture isolate. So, and so these are all the various automated equipment. So compared to the conventional, the time is very much reduced and easy to perform, and less strenuous work. So, and more accurate results. But the limitations are costly to establish the equipment, and so even most of every database might not cover every anaerobic pathogen, but still it is useful. And most of these automations use the miniature biochemical systems, which are a compact standardized panels with micro wells preloaded with the dehydrated substrates. So after inoculation and incubation, so there is a metabolic reaction takes place, and the color changes or the fluorescence changes indicating a specific biochemical reaction happened. So, and the change in colors and fluorescence are interpreted manually or even by the readers, or compared with the database. So this is a faster and reproducible system, and where small reagents are required, small volumes are required, so economized over a time. And so this is definitely useful system to adopt. But remember, even many times to conclude the reactions in the automation also, if it is not 100% pure growth or insufficient growth to put up the miniature to load the wells, so simultaneously you always keep in hand the gram stain, the colony morphology, and the aero tolerance, like some data, so that which you can interpret at the time of confirmation of the pathogen. So Vitek2 system, which is widely used, and nowadays almost most of the hospitals or the medical colleges, the laboratories are procuring this. And it has a wide variety of anaerobic bacteria and various biochemical tests and ANC card, which is meant for the diagnosis of anaerobes. And most of the times the ID comes within 6 hours, and it provides as a % probability. So the values close to 99 indicates the closer match to the given organism. So apart from these, there are the traditional or the conventional ways for determination of volatile acids and even non-volatile acids using gas liquid chromatography, which is a practical, inexpensive method, mostly followed. The basic principle is the metabolic pathways of the bacteria are genetically stable and conserved areas. So this the diagnosis of these particular metabolic products is more confirmatory for the detection of pathogen. So the metabolic products of carbohydrate fermentation or amino acid degradation are taken up for GLC, and especially volatile fatty acids and non-volatile organic acids. And non-volatile has to be methylated directly; they cannot take up. And so even this GLC is tried even directly on the patient samples also, but most of the times it is helped only in the presumptive clue, not the confirmatory. And to perform the GLC, and peptone yeast extract glucose broth is preferred for analysis sake. And this is another extension of the GLC, MIDI instant anaerobe for Sherlock. This is the name of the that GLC equipment, where microbes are analyzed by the fatty acids. The advantage is that the quantity, the cell quantity, and the reagents and everything goes very minimal. Even micrometers, 2.52 grams of colony and 250 microliters of the reagents. So such a minute quantities can be used in this, and it uses the Agilent technologies, analyzes the reports. So the results come in less than 15 minutes. So anaerobic blood cultures. So this is actually, we are all now, everybody is having the automated blood culture system. So Bactec, where this lytic, 10 anaerobic, or F is the facultative. And so this is here the headspace is filled with nitrogen and carbon dioxide. So for anaerobic specific bottles, there won't be any oxygen. So any kind of enhancement in the CO2 in the headspace, the sensors, the optical sensors will detect the change, and that will be fluoresced, and the positive signaled bottles will be taken out. So, and this is again Bacterialert FN bottles. Here we call them as. So usually recommended is both facultative anaerobes and obligate anaerobes. Both will be detected. So you prefer FN and FA bottles together. So even facultative anaerobes also can be cultivated. So 2 bottles at a time are recommended, 10 ml each. So this contains the BHA broth with EcoSorb, which is absorbs all the antimicrobials, all the inhibitors present in the sample. And so next to the cultures and all that, so the role of serological methods or the immunological tests are very, very crucial here, because as the difficulty in cultivation of the anaerobes, the process, the everything. So definitely this has a very futuristic hopes in the diagnosis of anaerobic bacteria, but still it is not that much used. Only for commercial kits now widely available for the detection of Clostridium difficile toxins in fecal samples, the antigens from fecal samples, And even glutamate dehydrogenase antigen is also a screening test for detection of C. difficile in the stool. So it helps to differentiate carrier state from the active state. So if GDH is positive, then we can choose for the toxin kit to detect whether it is toxin producing or not. So come to the role of molecular methods. Definitely, this is the now after post-COVID, everybody is speaking so in a scenario, especially in the government sector, all the authorities are behind these nucleic acid methods. Detecting the nucleic acid from the clinical samples, definitely they are rapid, accurate, and we can detect the direct virulence genes, or even we can detect the resistance patterns, everything, because the conventional has its shortfalls or the limitations. So these are the wide variety of molecular methods, PCR, multiplex PCR, real-time PCR, and 16S rRNA gene sequencing. So where, because rRNA is the highly conserved region, which helps in accurate, more specific in detection. Fish is there, and we can directly visualize the bacteria from the materials, but it has a limited probes. So microarrays is another method, because the fixed probes are hybridized by the bacterial DNA. High throughput detection provides, but it's a costly phenomena. And come to the sequencing, NGS and WGS. This NGS of the metagenomics, it is actually useful, because being a polymicrobial, it can detect all the microbes. It can sequence all the microbes that are present in the samples. So, but the thing is, bioinformatics expert is needed to discriminate all the data. But how far we can apply the clinical significance is again a point of a question. And WGS, we can a specific pathogen can be sequenced, and we can study for epidemiological and presence of any resistant genes, etc. But in most of these are not being routinely used. But yes, PCR can taken up for direct detection of DNA from the clinical materials. So when come to the antibiotic susceptibility testing, so actually for anaerobes, usually, as we mentioned in the first slides itself, the clinicians are just seeing the clinical picture suggestive of anaerobic infections with a putrid odor or a necrosis. They simply jump over to the selection of metronidazole or some other antibiotics, including a broad spectrum for control of the infections. But still, because of the developing resistance patterns, we need, may not be routinely, but for certain conditions, we need to perform the AST for anaerobes. So the recommended method both by the CLSA and UCAST is the agar dilution, which is a gold standard. And this diffusion is not recommended by both the organizations. So the media used for conventional testing of AST by either for E-strip or agar dilution is the Brucella blood agar, Wilkins Chagrin agar, Fastidious anaerobic agar, and Muller-Hinton agar, which is modified. That means supplemented with shea blood, vitamin K, hemin, and all this. So, but whatever media, any media, anywhere intended to use for anaerobes, it should be reduced. There should be reducing substances to remove the oxygen, and it should be processed in the anaerobic environment. So routine AST is definitely not recommended, because it's time consuming and not necessary, but limited used when your culture is impure, either from the blood or from the bone or from the CNS or any kind of sterile sites, or if the infection is suspected of anaerobe and not responding to the treatment. So then you can definitely choose for antibiotic susceptibility testing. What are all the various antibiotics used is a list of antibiotics. So we can put up those. And remember that anaerobic gram-negative bacteria are a significant reservoirs of antibiotic-resistant genes. So because especially in the large intestine or a part of the normal flora, they also play a role in the quorum sensing. They share the resistant mechanisms like efflux pumps, enzymatic inactivations through horizontal gene transfer. So they are the silent players in transfer of the resistant genes. So finally, to conclude, being a microbiologist, for taking up an anaerobic workout and to make it standardized, to make it a routine, or to make it useful in the patient care, we should always work closely with the clinicians, and because this will enhance the quality and clinical relevance of the specimens which are received for processing of anaerobic cultures. But remember, there is a role of clinician very much here. Always the clinical suspicion is very important. They should provide all the clinical clues to us, and the specimen collection is important, and meticulous collection we have to train them, educate them, and provide the time to time knowledge, and essential patient information definitely they have to supply. And being a microbiologist, we have to provide the timely diagnostic information and to help in the treatment decisions and ensure appropriate antibiotic use and minimum development of the resistance. So these are all the various references I have used to prepare this content, and thank you all.
Thank you so much, ma'am. That was really, really wonderfully explained. You covered all the way from the basics, the foundation of anaerobic bacteriology, to all the way, and then the good practices in processing of anaerobic samples. And I request everybody to mute, please. Thank you so much, ma'am. It was really good.
Thank you. Thank you.
We'll open up for questions, ma'am.
Dr. Vijendra, thank you for coordinating, and Prashanti, that was a very nice presentation. Dr. Jyoti here.
Thank you, ma'am. Thank you. Thank you so much, ma'am.
I was also
still remember,
thinking,
madam, you means we were all there that time when we closely observed this work.
I was a guide for him, and
Yes, ma'am.
I remember how excited we were when his first isolates came up.
Yes, ma'am.
Because I really appreciate your reiteration that bedside inoculation of culture has to be done. Remember, Prashanti, he used to always go to the OT, brain
Yes,
abscess.
ma'am. Yes.
He
He
used to go
used
to
to
OT
be
and
there
collect.
on the bedside, I mean, and the operation table side, whatever the abscess comes directly, he used to inoculate in the thioglycolate broth and bring, and he procured.
He used to use that laked blood agar.
Yes, ma'am.
And
Vishnu
both
procured
reduced.
media from Hardy Diagnostics. He imported from the US and he paid the customs, everything, madam.
Correct, I remember.
Amazing and hardship he has done.
Correct. So one more thing I just wanted to add, Ma, that though antibiotic sensitivity is not really recommended, but during his study only, we saw one case of metrogyl resistance. And he was using Brucella blood agar, I think, for AST. So,
Okay,
yeah, it is correct. I mean, not necessary to do always. And you have explained very clearly clinical suspicion, the importance of having pre-reduced media, the importance of transportation, and very nicely summarized all the media, all the tests available, all the methods. Nice to hear you, Prashanti. Congratulations.
Thank you.
Any questions, please raise your hand and go ahead.
So I think if the organizers want, I can share this PPT to you. So organizers may share to the participants.
Yes, ma'am. Surely, sure.
Actually, whoever is asking for the PPT should send one summary to Prashanti ma'am.
Yes.
See, just having the PPT is one thing, but going through it and summarizing it is another thing. So it's good that interested PGs are there and they have joined, but it is also important they go through it. It is really a beautifully done work.
Yes, ma'am.
Ma'am, there are few questions in the chat box.
Yes.
Why disk diffusion is not recommended for AFST?
Yes. So, because the anaerobes, the cultivation and the duration probably could be difficult, and the metronidazole is the one which is the for it responds to it kills the anaerobes and which provides the uh, as a diagnostic test, as a identification test they are using. And the CLSI is not recommending this. Only the agar dilution methods are recommended. Probably the standardization and the anaerobic cultivation may be the difficult. So as such, they didn't give the particular reason. So what I understood is this is the possible reason. What else? So it's clinically significant anaerobes fail to grow even in the ideal anaerobic systems. So I think obligate anaerobes are very difficult to cultivate. As I mentioned, so there are so many factors which should be meticulously taken care. So, and then your isolation definitely will improve depending on the media and the transport and the duration of exposure and what environment you are maintaining. So there are certain anaerobes which maybe died even in less than 10 minutes, exposed to the air, kill them in less than 10 minutes. So because everything should be ready in your hand, then in a matter of quick minutes, you have to transfer or maintain the streaks and put back to the anaerobic environment. So somewhere this fails, may get killed. Or still some non-cultivable anaerobes are there because I don't come across this non-cultivable anaerobes anywhere. And management of metronidazole resistant cases. Clindamycin is the recommended one, most of the cases. So metronidazole risk is not much reported. Maybe as just madam mentioned, some may come across. Most of the labs do not have the required media equipment for anaerobes. What is the bare minimum done for the clinical suspects? So that's what I suggested. So having the material, the pouch and the gas pack system and your regular blood agar or partially helps for the faculty to anaerobes, may not be the obligate anaerobes. And if you are able to procure the pras media, really difficult, but you can do it, you can get it. So thing is more important is your collection and transport. So if that goes wrong, even whatever efforts we made in the lab is also get wasted. Most of the times when collecting samples in BHI, thioglycolate, anaerobic cultures, not success. So only these two are not sufficient. So we'll improve sample collection from OT bedside. Definitely. Bedside collection, the two abscess pus or the tissue necrosed material after thoroughly cleaning the site. So these are all the materials definitely help improve the isolation. So we should not hesitate, as Jyoti madam mentioned, that post-radiate used to attend even in the emergency surgeries also. He used to go for CNS pus to collect from the OT directly. So that kind of definitely is difficult, but that dedication definitely helps. Metronidazole to do routine. Now they are asking for metronidazole sensitivity to do routinely. So routinely means I don't know, because isolation itself is a challenging, and performing metronidazole actually is a indicator disc which we can put in the primary streak line itself. So if the surrounding colonies around the disc are cleared, then possibility of anaerobe you can suspect and you can give a clue. But sensitivity performing is not recommended. So if the clinician strictly wants that, then we have to choose the agar dilutions or any automation. But I don't know, automation is the disc widely available or not. Raising resistance in Bacteroides fragilis group. How should laboratories guide clinicians when AST facilities are limited. And as on now, anaerobic bacterial resistance patterns, not like our regular multi-drug resistance patterns of our regular GNBs. So what the literature says is, so the regular metronidazole, what the clinicians are using, and some broad spectrum antibiotics, even the meropenem, whatever they are using, are still working for anaerobes. Surgery colleagues suspect gas gangrene, want to do anaerobic culture. Collect in thioglycolate, inoculate, and basic media. It's very difficult. It's very difficult. So yes, you may partially observe based on the color or pigment production or any kind, but just morphologically, a presumptive diagnosis you may do, Dr. Swarup. So a presumptive diagnosis, a clue you may give, but not be the confirmed isolate. So I already mentioned in the slides that with the samples. So already always prefer the deep-seated pus or tissues, so which are a bit away from the mucosal sites. For gas gangrene, clinical and gram stain is good. Yes, it provides a provisional clue, as Jyoti Madam said. Yes. Branching filamentous organisms in success. Only direct gram stain is achieved, but not anaerobic culture. Filamentous organisms means actinomyces and success. Only direct gram stain is achieved, but not anaerobic culture. We tried BHI and thioglycolate. So maybe this DHI, thioglycolate RCM may not be sufficient. You need some means the blood agar based medias or kanamycin, vancomycin agars to support the growth. Approach to reporting anaerobes isolated from polymicrobial infections. Single most important factor that improves anaerobic culture. Yes. In routine practice. Dr. Pallavi, yes, true. Because everywhere, whatever we are going through the literature, so everywhere they are mentioning the polymicrobial nature is a big limitation in the isolation or clinical implication of the anaerobes. So I think we have.
I'll ask the last question, ma'am. With regard to blood cultures, now it's recommended you send up around 3 sets of blood cultures for maximum isolation. So what clinicians are telling that they would want to send aerobic plus anaerobic set.
Yes.
Now, how much is it recommended that an anaerobic set of blood cultures is included in every patient?
So actually, they are mentioning this FN bottles not only supports obligate anaerobes, but also facultative anaerobes. So for that reason, they are recommending that FA bottles, along with FA, FN also. So probably in case of any kind of fastidious organisms which needs CO2, that will grow or multiply in these bottles. So FN bottles, I don't know because I never worked on this. I never observed that. But the meningococci or such isolations are improved. The pneumococcal or any so the fastidious organisms isolations are improved in the FN bottles. In that way, it helps, which maybe every time not possible with FA bottles. So in that way, it is helping. So this joint cultures of 10 ml, 10 ml each in these 2 bottles will improve the isolation of these fastidious facultative anaerobes. So true anaerobes could grow, but I don't know about that knowledge. I never observed that.
Okay, ma'am. I think we'll wind up the session, ma'am. Thank you so much. Thank you so much once again. It was really very nice, very informative, and thank you so much for accepting to have this session.
Thank you, Vijender. Thank you, one and all, for your patient listening. And I'll share to Dr. Vijender, and maybe he will share to the site and through groups.
Done. Fine. Thank you
Thank you.
everyone for attending. Thank you, ma'am. Bye. Good night.
Good night. Good night, all.